Okay, so I have 11:35, which is when we're supposed to begin the next group, which will be SLF Phenology and Forest Impacts. So we'll have three presenters. So our first presenter is Dr. Melody Keena, who's with the USDA Forest Service. And if you're all set to go, Melody.
I'm getting set now.
Yep. Okay, yeah, that looks good.
Everybody should see it now.
Yep.
Okay, so I'm going to cover a little bit of what's already published, but more of the study that was done last year and some studies that are going on as we speak, all concerning spotted lanternfly phenology aspects. And these are the collaborators on different parts of the work at Rutgers, George Hamilton, and the graduate student, Devin Creederman. And at the Forest Service, Talbot Trotter is working on the modeling that we're doing. So these are the studies that we have worked on or are working on. The two in blue are already published. The temperature requirements for egg hatch effects, the effects of photoperiod on hatch timing. That one's not published, but it is complete. And in reference to some of the questions yesterday, the SLF eggs, if you collect them in the fall, shortly after they're laid, they will proceed to hatch at 20 degrees, 15 degrees, or 10 degrees without any temperature going lower than that. So that's part of what goes on with the SLF. And the temperature effects on the nymphs and the upper and lower thresholds and survival at different temperatures, that's also published with degree day estimates for each of them. The yellow ones are, the data is complete. It's all been collected, but they're not published yet. Effects of hosts on the development. We looked at several different hosts and determined how many days they spent in instars on the different hosts and the survival on the different hosts for each of the 4 instars. And this last year, we looked at effects of cold snaps and heat waves on the lanternfly in order to be able to adjust phenology models to account for variable temperatures and to confirm the thresholds that we had estimated previously. And we also collected the entire life cycle using a mimicked Napa, California temperature regime and light cycle and humidity. And what we're working on right now is the potential for local climate adaptation, and Talbot is working on an agent-based phenology model, and I'll talk a little bit more about those toward the end of the talk. For those that aren't familiar, these are the 2 papers. They're both in environmental entomology, and they are both out. Although the bottom one doesn't show it, it came out in 2021. So for the heat wave and cold snap study, part of that was spurred on by this paper that was published not too long ago that showed that as you move north and as you move south, that the favorability of those climates for spotted lanternfly declines. And so we wanted to look at what's going to happen if they do move into those different regions in terms of how are the nymphs going to respond. So this is our developmental data for the nymphs. And what we chose to use were temperatures that were slightly outside the range at which they molted for constant temperatures. So we used 5 degrees C as a lower cold snap temperature, and we used 35 and 40 degrees C as the heat wave temperatures. We did have in the 3rd instar, a couple of individuals were able to molt at 35, but not in any of the other higher instars. So we're looking at cold snaps for the first 2 instars and heat waves for the second 2. So this is the setup of the study. The cold snaps, we're comparing a constant 15 degrees C treatment against a treatment that averaged out to 15 degrees C It had 8 hours of 5 degrees C and 16 hours of 20 degrees C, and it was an instantaneous drop in temperature, so a very sharp drop. For heat waves, similar setup, they averaged out to 25 degrees C, 8 hours of 35 or 6 hours of 40, with the rest of the time at 20 degrees C. We also ran a second control of 25 degrees C for both of these parts of the study. The egg masses that we collected to use in this study came from 3 different areas near Philadelphia in what is the 7B climatic zone, and in New Jersey from 6B close to a 6A area, and from Virginia in a 6B area. We hatched the nymphs, and as soon as they had hardened off, they went into cages. The small cages were used for the data collection. Large cages were used to rear nymphs to particular instars other than first, so for seconds, thirds, and fourths, and then they were moved to the smaller cages for the actual data collection. And part of this study, we also looked at the hatch of those egg masses from the 3 areas. So the previous year, the paper on hatch is all based on the purple, which comes right down through the middle here with an average of 92 days to mean hatch at 15 degrees C constant temperature. The New Jersey ended up with an 85-day mean hatch, and one of the Pennsylvania ones ended up with 101 days as the mean hatch. So we were seeing some differences in hatch profiles at 15 degrees C from these 3 different collections that year. For the survival, I'm just going to show you a couple of instars. For the first instar, when you gave them the cold snap, which is the 25 treatment, the mortality was higher than at the 15-degree treatment and the 25-degree treatment. In the third instar, it was interesting that the 40-degree heat wave treatment had very high mortality, and very few were able to molt to the next instar. But the 35-20 treatment was better survival than the 25-degree control treatment. In the cold snaps, we saw that the number of days that they spent in that first instar, but not the second instar, increased as they were exposed to the cold snap, the 5-degree treatment. And in the second instar, nymphs initially went, well, both instars. Initially, the nymphs would go into a cold coma for a few minutes in the first few days they were exposed to the treatment when they were moved initially to 5 degrees C. But after a few days, they stopped going into this cold coma. In terms of weights, for first instars, the weight of individuals that were reared in the cold snap was lower than those that were reared at the 15-degree treatment. And for the third instars, though, so the second instars that became third instars, the weights of these new third instars were actually higher for the cold snap. And we think that that may have something to do with only the most robust ones were able to survive that treatment. For the heat waves, number of days in instar increased with the heat wave temperature at for third instars as the high part of the heat treatment got higher. So did the mortality, and so did the mean time in instar. We had trouble with fourth instars, not enough chamber space, and they don't like to be in the small cages as well as the other instars, so we didn't collect as much data there, but it looks like for fourth instars, the same thing was happening, that they spent longer in the instar. And for fourth instars in particular, and third instars also, any that were able to molt at the higher temperature, the 40-20 combination, they didn't sclerotize properly. They looked light and fragile. So in the South, places where 40-degree temperatures may occur it probably won't be very good for the SLF. Even though they're able to molt with these temperatures outside of the threshold area, their vigor is affected. And the weights also, you can see, are affected. The other piece of this that was interesting is taking the two control temperatures and just looking at how the different populations fell out. The coldest location from which the egg masses were from was the New Jersey one, and the warmest was the Pennsylvania one. And their response to 15 degrees fell in that same climatic regime. So the coldest ones were able to complete development faster than the ones from the warmest location. But at 25 degrees, there was no difference. Second instars, there's some trends, but not a lot of significant differences there. And third instars, there were some differences at the 25-degree side of things, with those that were from the warmest location going through faster than those from the colder locations. All of these things taken together, the hatch profile differences and these different responses to heat waves and cold snaps, seem to be indicating that there might be some local climatic adaptation going on. And so we felt this merited some further study. So just to summarize, for the cold snap and the heat wave conclusions, nymphs can develop, although survival may be lower at temperatures above and below the developmental thresholds in an alternating temperature regime. And nymphs from different climates may be adapting to develop to cooler or warmer temperatures in the zones where they are found. And this is the funding source and the people to thank for that. So moving on to the other piece of last year, this study was also a very interesting study, looking at the phenology of the spotted lanternfly under the Napa, California regime. We took the last 10 years of temperature and humidity data and created weekly averages, and then the chamber was set to have the minimum temperature at the average minimum temperature for that week and the maximum temperature at the average maximum temperature for that week. And then it would ramp between those two so that it was mimicking an outdoor regime, with the light cycle being changed on a weekly basis and the humidity also. We followed them through. We put the same eggs from the same locations as was used in the cold snap heat wave study at this regime. And they hatched here, with the first hatch occurring on Julian date 102, which is about April 12th, whereas the 15-degree hatch, just for your reference, occurred over here, same sets of egg masses. And then they proceeded through the different instars, and we saw that males came through first and then females, and the first eggs laid on Julian date 284, October 11th. Probably a little later than would be natural. It's hard to rear adults and get eggs in the laboratory, so that could be suspect. But this data set is going to be available to do validation of models that are being developed. So the local climatic adaptation assessment that is underway now collected populations from 20 different areas. Most of them were collected in October. A few where I couldn't get to were collected a little later. But you can see we have them from Connecticut, New York, across Pennsylvania, in Delaware, New Jersey, some shipped in from Virginia, some from Maryland. And the intention was to collect populations from the different cold hardiness zones, and that's what's being depicted here. So we tried to collect 3 or 4 populations from each of the hardiness zones from 7B, 7A, 6B, 6A, and we think we may have even moved into the 5 a little bit. And so the hatch at 15 degrees C, 15 is a very good temperature to spread out the hatch and see any differences that might be present in the population, either within an egg mass or between egg masses. And I've color coded all of these populations by cold hardiness zones. So the populations from the 7B zones are here in red. The ones in here that are in the yellow are the 7As, the 6Bs are here in green, and the 6As are traipsing across here in blue, and the population from the coldest site is right here. So it looks like, although we have to still correct this for the possible cool temperatures they might have been exposed to between egg laying and collection, it looks like that those populations in the warmer zones are hatching slower than those in the cooler zones. And it looks like there may be some indication that egg masses are being laid at different points in time, that there may be gaps in time when egg masses are laid. And I think that's what's going on with these plateaus that are going on here. The other part of it is rearing the hatch at 15 degrees C for the first instars and 25 degrees C, similar to the cold snap and heat wave study. This data is just coming in. As we speak, I updated the 15-degree data this morning. What we do see is that the time in instar does vary between populations. It doesn't appear to be varying in a particular pattern right now, but I'm thinking that there may be some patterns that will emerge eventually when we look at the size of the population from which the egg masses were obtained, the egg mass size, which may also be tied back to how big the population was or how long it's been there and how much they've impacted their resources. There may be some patterns with reference to that. But again, this population here circled in red is the same New Jersey location as the year before. Egg masses were smaller this year, population size was still up, but its ability to develop faster at cool temperatures remains constant for that site. These other sites are just starting to roll in, so these are not the final averages for time in instar, but this will be very interesting to see how this all plays out. And these are all the different people that helped with collecting those egg masses for this year. So a lot of people to thank. And then just a real quick tidbit on what Talbot is working on. The model that he is developing will be an individual based model. So the individual will go through each instar based on the percentage of individuals that go through at a particular rate in an instar to make it to adult. And then there'll be like 100,000 different individuals to get a percentage that should be becoming a particular instar on a particular day. So there's a lot of steps in it, and he's still working on the egg piece of it. The eggs are a little bit more complicated, but it's beginning to look like from these two data sets, the Napa data set and some others, that the eggs aren't going to hatch outdoors until the minimum average temperature exceeds their developmental threshold. And that may be why we're seeing some differences from year to year. If you just use a straight degree day model, you're not going to predict that hatch, but there's more to be done on that to finalize that. And I will stop there and stop sharing and answer any questions.
All right, thanks, Melody. Yeah, I don't see anything in the Q&A section this time. I know there's a lot of good chat, a lot of good things going on in the chat, so you might want to just take a look over there, Melody. But yeah, I don't see anything in the Q&A section.
All right, thanks, Greg.
Yeah. All right, so we'll move on to Houping Liu, who's with Pennsylvania DCNR.
Okay. Good morning, everyone.
Hey.
Thank you for the opportunity.
Yeah. Oh, and we can see the slides on the left. Houping. It's
okay. Yeah. You want me to do the?
Yeah.
Okay. Is that around?
Yes. Yep.
Okay.
Thanks.
Great. Thanks. We have a couple of projects going on. Today, I'm going to be focused on the infrared thermography as a potential detection tool for the lantern fly adults. So first, there are a couple of, back to the high school physics. the loss of electromagnetic radiation. The first Planck's law said all objects with a temperature above absolute zero, that's 0 Kelvin, will emit radiation with the amount of radiation dependent on the temperature. Basically, the higher the temperature, the more radiation the object will emit. And the Weis law says the peak emission is inversely proportional to the temperature. That means the higher the temperature, the smaller the wavelengths for the peak emission. And the Stefan Boltzmann law says the energy emitted by the object is proportional to the 4th power of its temperature. And Kirchhoff law says the object's emissivity is equal to its absorptivity in the thermodynamic equilibrium. That means, like, for example, for the idealized black body, the emissivity will be equals 1, and the opposite will be the white body with the emissivity of 0. So everything else is between 0 and 1. So believe it or not, all the objects will have this whole spectrum of the electromagnetic spectrum, from the radio wave to microwave to infrared, ultraviolet, X-ray, gamma ray. It's just that, like, for example, the humans, the gamma ray we emitted is very, very small, so you cannot detect that. So based on this, the spectrum, for humans, we can only see these lights between 400 to 700 nanometers, but the infrared spectrum is between 0.7 to 1,000 micrometers. So the infrared thermography is using the specially designed cameras to detect the long wavelength infrared at the spectrum of 8 to 14 micrometers to form a thermogram based on thermal heterogeneity. Like for the surface of any object, the temperature will be different. So here's the example of a cat that has a higher temperature on the eyes and mouth, ears, but lower temperature on the fur, on other body parts. The color is artificial, it's not real. So how can you tell the normal picture from the infrared thermography? Just this one, this is the back of the TV with the Wi-Fi radio tower. Pay attention to the lights you can see right now. And when you put it under infrared thermography, you do not see the light. And for this particular white hot color palette, the center with of those two objects are the ones has the highest temperature. If I change it to black hot mauve, and the center will turn to black. That means the highest temperature part is in black. And so with hot red, so the highest temperature place will be yellow. And this one under red monochrome will be like kind of white. And the rainbow is red. So the highest temperature for sure of the object is red. And ultramarine is the same. And violet is kind of white and yellow. And it's sepia. Depends on the cameras you have. I think the color palettes will be different a little bit, but you can use any of those to indicate the temperature difference of the objects you are testing. So we've been wondering about this like since 2018. See, he said, because we know that infrared camera has been used by widely used by firefighters, by law enforcement, by even medical imagery. But in entomology, I think most of the applications are limited to thermal regulations in the laboratory. We were wondering about this since 2018. Say, hey, what if we can use them in the field to detect some of the insects? And this just happens. One of our co-authors, Ross, is an avid bird watcher. He actually went to one of the parks near his home at night to do some bird watching, and he captured this picture. This is a maple tree. It has a lot of lentifera on it. He didn't know what it is at the beginning. Just to see each white spot represent a lentifera adult, and he later found out this is actually the insect. So this is two of the close look at the two of the adults on the Chinese privet. And as you can see from this picture, you can even see the legs. You know, the very, very bright against this dark background, and of course the temperature gradients from the head to the tail of the body to the end of the body. So after a couple of preliminary studies, and we designed this simple experiment in one of the local parks. We found this 3 heavily infested red maple trees. It happens to be in a triangle, equilateral triangle. So we put the cameras in the middle, in the center of those 3 trees, and the distance between the camera and each tree was about 4 meters. The trees are about 15 inches in diameter. It's a large tree. There are large trees like 50 in height. So we checked on the 2 trunks, 100 centimeter trunks on each tree, because there were a lot of adults on it. So we checked them every 30 minutes between 4:30 to 7:30 in the morning. And we count the adults with the flashlights, and then we use the infrared cameras to count how many are lighting up under the versus not, those ones not light up. So many of them actually lighted up, just like what Ross observed in the other trees before. So in this picture, you can see a couple of them light up like that, but they're also adults. You can see that over here does not light up. And for this one, for this picture here, you can see the clear temperature gradients from the head to half of the wings. So in terms of the detection rate, from 4:30 to 6, about 40% of the insects on the trunk were detected by the camera. So the detection rate decreased from 6:30 to 7, about 35%. By 7:30, that's when the sun comes up, I think interfered with the detection. So they dropped to less than 10%. And we also found, we used two different cameras, and we found there's no significant difference between the cameras. Of course, our data, our sample size was kind of small. So this phenomenon was also confirmed by another scientist, Dept. He did a study, he did an observation in New Jersey between 2020 and 2021 on the spotted lanternfly. Here's what he saw, he published it in this journal. Although his picture does not have the detailed temperature differences between different parts. And he also found the same phenomena on a different lanternfly species in Vietnam, this one. So it does seems like the usage of the infrared camera is not limited to a lanternfly, a spotted lanternfly alone, can be applied to other lanternflies. So as we all know that insects, their body temperature depend on the environment. But many insects can actually adjust their temperature in the environment based on like the ectotherms, like these wood ants can increase their temperature by basking, and the endotherms like honeybees, they can actually increase their temperature by through some biochemical processes during flight and other activities. So we all know that insects with the piercing striking mouthpart have this structure called symborium palm and dilator muscles to operate. This is a picture of the McLean and Kinsey. It is widely believed that for the insects feed on the phoron, they don't need to have a well-developed pump because the phoron was positively pressurized. They just need to insert their mouth part, and the fruit will actually automatically flow to their mouth. On the other hand, for the insects that feed on xylem, they do need to have a strong pump to pump the liquid out. So the question right now is SE a pure phoron feeder, or they can feed on both the phoron and the xylem? So that's important because that could help us explain why the spotted lanternfly can be detected by the infrared camera, because it is widely believed that active feeding, just like all the lanternflies, can create some kind of generate some kind of heat signature. And here's a publication from 2007. They actually measured the honeydews ejected by the feeding adults. There are several times of this body mass per hour. Think about that. And another one is possibility is how about the circulation of the hemolymph from head to other part of the body? Does that create thermal heterogeneity that's detectable with the infrared camera? I think earlier on you heard Julie talk about the EPGs. I think maybe we can combine those study with the EPGs and the measure of the infrared camera. This is just a diagram from Dr. Bacchus, how it works. So just a little bit additional information about lanternfly. We all know the meaning of lanternfly. So in the early 17th and 18th century, a lot of European travelers, explorers, they found some insects that can emit light in South America, Central America. That's not until German naturalist Marianne, she actually described the luminous nature of the peanut-headed bug, Bogora lanteralia. And Linnaeus actually popularized the lanternfly term when he established the genus Bogora, used the peanut-headed bug as a type species. However, there's no record after that. Nobody recorded any of the lanternfly bug insects actually have emitted any visible light. So it's created one of the mysteries, entomological mysteries in the history. So here's the depiction of one of the artists figure produced, let's say, when Marion was presented with the box of the peanut-headed bug. She was amazed that the lights is so bright that she can read a book under it. So later on, some scientists believe that Marion may mistaken the Houping click beetle, which actually has the lighting organs on the thorax and one at the tip of the abdomen. She actually, maybe people think maybe she made a mistake, by looking at this bug and it says it's a peanut-headed bug. However, there is a very good artist. He actually made this picture, with this drawing with exact depiction of the insects. So I kind of doubt she will make that kind of mistake, but I don't know. So the bottom line is that spot lentiviral does not produce visible light, but it can be detected with the infrared camera. So for more information, you can look at our article published last year at the Journal of Pest Science. So I would like to thank Matt, Tim, and Heather for logistics support, Charles, Julie, Holly for the discussions we had on the pond, and the conservation district, the township, and the Marsh Creek State Park for study site access, and the Department of Conservation and Natural Resources in Pennsylvania for the funding. Thank you.
All right, thanks, Houping.
Yeah.
Yeah. Thanks for that update on that. Yeah, I don't see anything in the questions and answers, but again, there was a lot of things going on in the chat, so you might want to just take a look over there too.
Okay.
Okay. Oh, wait. Oh, there is one here that came up just now. It asked, there was a question, do SLF nymphs and egg masses show up in heat mapping?
No, we tried that. No, did not show up. That's why we think it may have something to do with the feeding of the adults.
Okay. All right, thanks. Yeah, I know Julie Urban had talked a while ago about the when this was first coming up, and about so the endosymbionts and whether that was what was causing some of the heat signature.
Mm-hmm.
Yeah. Okay, all right.
Looks like a couple new questions came in.
Oh, sorry. Thanks for catching that, Brian. Oh, and what time of summer were the glowing SLF seen?
At what time? I think they feed all the time, like 24. Of course, we didn't follow 24/7, but when we get there, like in 4 o'clock in the morning, I think they are feeding. Some of them are feeding. So the problem is that we don't know exactly whether they are feeding or not, because you can see their insert their mouth part in the tree, but we didn't know whether they are actually feeding or just sit there. The only sign we can see is like sometimes in the morning you can see the honeydews expelled from the tip of the abdomen.
And do you know what the dates were, or when that was, or what months that was in the summer?
We did that in October, I believe.
Okay, all right. Yeah, because I was trying to remember when those photos came out, if that was in September or if that was in October too.
Yeah, that was a surprise. I remember I was asking the question at our group meeting, and not everybody was convinced. I was not convinced that we can detect that because we have no idea. But I would just have the curiosity to say, what if? But then Ross had this picture. It's like, great. So we went on to plan the study right away. Yeah, we had to let the township police know that we are doing this in the park. They don't come. Somebody was doing something fishy like at 4 o'clock in the morning in the township park.
And then there is 2 questions that are I think about the same thing, asking if the SLF that showed up, were they male or female, or do both of them show up equally?
We did not separate that. That's something we can look at. Actually, we are planning something hopefully this year, as other speakers also mentioned. It's very difficult to read adults in the lab. So hopefully we can do something in the lab this year, see whether we can figure out some of those questions.
Okay, thanks, Houping.
Thank you.
And so our next presenter would be sorry, I think you have to stop sharing, Houping. Yeah, our next presenter will be Dr. Jennifer Chandler. You'll have to identify where you're from now, Jennifer. I'm sorry.
Yeah, no worries.
I know it was Westchester University before, but I know you're not there anymore.
Yeah, so I'm actually working with Westchester University now, and I'm excited to be here. Thank you. I want to talk about something a little different that came out of the work that my lab was doing in the forest, and that is differential escape of spotted lanternfly from two really commonly used trapping techniques, and then the use in linking counts based on that to forest characteristics. And so the topic, like I said, that I'm going to discuss primarily today builds from two areas of work being done in my lab. The first is from a study that was developed and implemented and reported on in collaboration with a couple of, actually several undergraduate biology students, and I was really excited for them. And this project stemmed from a question that came up as we were setting up and monitoring our forested research sites. So the bulk of the talk is going to be spent on that. The second shorter part of the talk will involve how we took this information and applied it along with some field data that we've collected to learn a bit more about possible correlations between spotted lanternfly abundance and forest characteristics. So first, I should give you a little background about how the idea for the first part of this talk came about, that is quantifying differences in the probability of escape from adhesive bands as a function of spotted lanternfly life stage. So a while back, my lab was out at our field sites, placing tree bands around our representative trees in order to get an understanding of the number of spotted lanternfly that were in our sites, or at least crawling up some of the trees in our study site. So we banded numerous trees in each one of our sites, and we noticed virtually immediately that not all of the spotted lanternfly that had countered the sticky bands were actually captured. In fact, many of the spotted lanternfly were able to walk right onto the sticky bands and then walk straight back off, even when fresh bands were being placed on these trees. So it seemed to us, at least anecdotally, that the ease with which spotted lanternfly escaped the bands varied based on their life stages, but we really had no idea, no real way of knowing this or quantifying this. So then as we were driving back from our multiple field sites that summer, my students noticed that many homeowners in the area of Pennsylvania we're in had wrapped their trees in bands also, but these bands looked different from what we were using because they were sticky bands made out of duct tape. And so we knew from field observations that commercially produced sticky bands were not always effective in capturing the spotted lanternfly. And we figured the same was going to be true about the duct tape, but we simply couldn't find any information about the effectiveness of either of these bands. And we further couldn't find information about how effectiveness may vary depending on the life stage of the spotted lanternfly. And we thought to ourselves, like, a better understanding of the probability of an individual escaping from a given trap once it's encountered is really important when we're thinking about population estimates or just looking at the counts of bugs on our trees. So my students and I developed a controlled laboratory experiment, to answer some simple yet relatively unexplored questions that, while not directly related to the forest measurements we were taking, like out in the woods, may still inform the work that we're doing there. And these questions pertain both to commercially produced sticky bands, which is what we were using in the woods, and also to duct tape, these homemade tree bands that we were seeing from the homeowners throughout Pennsylvania. So the first question we asked is, does the effect of adhesive type on the probability of escape vary based on life stage? And we predicted that commercially produced sticky bands were going to be more effective than duct tape across all life stages. The second question we asked is, does the probability of escape vary based on a spotted lanternfly's life stage, regardless of which type of adhesive band is used? And we predicted that the probability of escape across both band types would increase as life stages increased. Our third question dealt with, does the probability of escape differ between the two band types, duct tape and sticky bands, regardless of which life stage we were using? And we predicted that even when brand new, the duct tape would always be inferior to the commercially produced sticky bands. And finally, we kind of like broke that up and we said, okay, based on what we learned from the first 3 questions, does the probability of spotted lanternfly escape vary among life stages for each of those 2 adhesive types separately, so taking them into separate accounts? And we predicted that the probability of escape on commercially produced sticky bands will increase as individuals progress through life stages, but overall will remain fairly low. And we also predicted that the probability of escape from duct tape will likely remain high regardless of the life stage of the spotted lanternfly. So my lab and my students collected spotted lanternfly from field sites located throughout Eastern Pennsylvania. Due to the opportunistic nature of the study, we were not able to collect enough first instars to incorporate experimentally, but we were able to capture and test a decent number of second, third, and fourth instars, and also adults. And so individuals were collected in mesh bags, and you can see my students actually doing this in this image. And they were immediately transported back to the lab, where they were held in separate mesh enclosures containing fresh tree of heaven branches for a period not to exceed 24 hours before being subjected to testing. The other upper portion of a slab of wood, and this is a recreation of this here, was wrapped completely in a strip of either Webcoat brand sticky tape or professional grade duct tape. And that slab of wood was oriented vertically in a separate mesh enclosure. We chose to use Webcoat brand sticky bands, number one, because that's what we were using in the field, but also because another study had indicated that this specific brand was more effective in spotted lanternfly capture than both a clear packing tape and also a different tree band that was produced by a separate manufacturer. The duct tape that we used in this study was an industrial grade all-weather heavy-duty duct tape with a rubber-based adhesive. And this particular duct tape surpassed the adhesive values of many of the standard non-professional grade duct tapes that were being utilized by homeowners when we surveyed those homeowners. So admittedly, only one brand of duct tape and one brand of sticky bands were used in this study. And even though these particular brands represented some of the strongest adhesives offered in their categories, we have to take those results and interpret them with care. So for each trial, a haphazardly selected individual was removed from its holding enclosure and its life stage was recorded. And that individual was then transported into the testing enclosure and was released at the base of that wooden slab and allowed to climb upward toward the awaiting trap, either the Webcoat brand sticky tape or duct tape. Once that individual actually encountered the trap and an escape attempt was initiated, a timer was set for 2 minutes, within which time the individual could attempt to escape. And this 2-minute limit was established as the escape threshold based on preliminary observations that lasted for far longer than 2 minutes and that just seemed cruel in my mind. And all of these prior trials indicated that if a spotted lanternfly did not escape after 2 minutes, they were unsuccessful in ever being able to free themselves. Fresh strips of adhesive were utilized for each trial, and each band type, the Webcoat sticky bands or duct tape, were alternated between trials. And so log likelihood analyses were performed to answer the questions I had, with escape either yes or no as the nominal response variable, adhesive type, the commercially produced sticky bands or duct tape, as the first nominal explanatory variable, and life stage, either second, third, or fourth instar, and then adult as the second nominal explanatory variable. And so I'll talk about these tests as I present my results, just to make it a little more clear. But I used a 2x4 factorial log likelihood analysis to explore those first 3 questions I laid out, and then separate log likelihood analyses that focus on those data split between the 2 adhesive types for our last question. And so just as a reminder, that first question asked if the effect of adhesive type on the probability of escape varies based on life stages. In other words, does the pattern we see in the ability of these insects to escape as they get bigger vary depending on whether they're exposed to duct tape or commercial sticky bands? And what we found is that the effect of life stage on probability of escape did not depend on the type of adhesive band used. And you can see here, I hope you can see my pointer, for each life stage, the proportion of individuals that were able to escape was substantially greater when using duct tape than when using commercial sticky bands. So that pattern of escape probability as you move across life stages did not differ between those 2. So we then broke that apart to ask the question, does the probability of spotted lanternfly escape vary based on life stages, regardless of the type of adhesive that's used? And so we pulled all of our data across both adhesive types and focused in only on the life stages. And what we found is, yes, the probability of an individual escaping did depend on the life stage of the individual when pulled across both adhesive types. And so this figure is kind of interesting. I want to just describe what it's saying here, because I have a few figures that look just like it coming up. So on our y-axis, we have the proportion of individuals that either did or did not escape. For this particular figure, the x-axis represents the life stage of interest. The dark gray bars represent the proportion of individuals in that given life stage that were able to escape. And so, for example, for the second instars, this dark part of the bar represents the proportion of individuals that were able to escape. And I focus on the escape proportions, so I've highlighted these in color. But this light gray that you see above that represents the portion of individuals in that given life stage that were captured. So what we see then is the percentage of spotted lanternfly escaping the adhesives were lowest in second instar individuals. So across both adhesive types, about 49% of second instars escaped. There was similarity among the third and fourth instars in terms of the proportion able to escape. And then we saw the greatest proportion of escaping individuals in the adults, which was expected. So this tells us that the probability of escape, regardless of which adhesive type is used, is larger as life stage progresses, with a huge jump in adults. We then answered the question, does the probability of spotted lanternfly escape vary based on the type of adhesive band that's used, regardless of which life stage of the individual we're talking about? And so the data were pulled across every life stage, and the only focus was band type between duct tape and sticky bands. And what we saw is, again, yes, the probability of an individual escaping does depend on the type of adhesive used when pulled across all life stages. So in this figure, we're still looking at the proportion of spotted lanternfly that were able to escape. But now our x-axis is adhesive type rather than life stage. So what we see is that the percentage of spotted lanternfly escaping the adhesives were far higher in the duct tape. In fact, about 92% of all individuals that were subjected to duct tape, regardless of life stage, were able to escape the duct tape. That number was substantially lower on the commercially produced sticky bands. In fact, over all life stages, only about 47% of individuals were able to escape. So this tells us that when life stages aren't considered, duct tape is really inefficient in capturing spotted lanternfly. Almost every single individual that crawled onto duct tape crawled right back off. We still wanted to know what is the effectiveness of each of these band types separately in capturing the different life stages of spotted lanternfly. So for this set of analyses, we split our data set up and we asked, does the probability of escape vary among life stages for each of the 2 adhesive types separately? And so we ran separate log likelihood analyses to figure this out. And so the figures are going to be set up similarly to before, but this time we'll have one whole figure for sticky bands and one whole figure for duct tape. And the life stage will be across the x-axis. So you can see here, this first figure deals only with sticky bands. And what we're seeing is the proportion of individuals that escaped the sticky band particularly, and on the x-axis, our life stages. So this first study, where we focused only on sticky band, commercially produced sticky bands, indicated that life stage does influence the probability of escaping from these web coat bands. The proportion of individuals that escaped from the sticky bands increased, as you can see, as life stage progressed. But for instars, so life stages 2 through 4, we still had a pretty low proportion of individuals that were able to escape those sticky bands. But there was a marked increase in escape for the adult life stage. When we look at duct tape, we found that life stage does influence the probability of escape from duct tape. But duct tape is high, escape from duct tape is high regardless of life stage. In fact, you can see that the probability of second instars escaping from duct tape is about 73%. And that's almost as high as the proportion of adults that were able to escape sticky bands. And what's more, 100% of the adults that were subjected to duct tape were able to walk on and straight back off. Not a wing got caught there. So all of these data tell us that duct tape is almost always an ineffective option to combat spotted lanternfly, but that sticky bands, on the other hand, seem to be quite effective, particularly for those early instars and to a lesser degree adults. Of course, we have to acknowledge that field conditions, including temperature variation and precipitation and the accumulation of debris and insect carcasses on the adhesive surfaces, can influence capture rates. And because this experiment was very much a controlled laboratory study, it really represents the best case scenario capture rates. And so when applied to field data, it will likely be a conservative estimate of the number of spotted lanternfly that are able to escape the bands. Excuse me. And so now I'd like to transition to the second part of the talk, which is just a very quick example of how we may apply the escape proportions we learned about in the first part to real catch data and further explore links of spotted lanternfly numbers to forest characteristics. And so in 2019, my lab began working to quantify the characteristics of the forests where spotted lanternfly infestations were seen, and to begin assessing how spotted lanternflies might affect the forest system. This latter part is still a work in progress. So this involved setting up FIA-style plots, which you can see in this diagram, where we quantified numerous characteristics about the tree layer, about saplings and seedlings and invasive plants in the forest. And for just the remainder of this very quick talk, the focus is going to be only on the tree layer. We collected all kinds of data on the trees within each subplot. I'm only going to talk about 2 of those pieces of information here, tree species and DBH. But we have the same types of data and the same types of correlations for height and crown class and crown dieback and damage class, live crown ratio, a lot of different variables. So we banded trees near each of our established forest plots or in our established forest plots to get a better understanding of the general amount of spotted lanternfly that were crawling on the trees in this area. The number of trees that were tagged per site varied based on tree availability, as did the total number of days that sticky bands were left out across the entire season for these trees. Sticky bands were removed and replaced approximately every 2 weeks during the season, and spent bands were brought back to the lab, and instars and adult spotted lanternflies are tallied. So our sticky band count data underestimated the quantity of spotted lanternfly that were traveling across those bands in the field on a daily basis, since not all spotted lanternfly that encountered the band were actually caught. So for each individual tree, we adjusted the number of instars and adults caught to a value we referred to, probably not so eloquently, as the estimated number of spotted lanternfly encountering sticky bands. And this just gives us a bit of a better understanding of the magnitude of individuals that may be walking across those sticky bands on the trees. So we use the proportion of individuals that escaped the sticky bands in that experimental trial to estimate how many individuals may have encountered that band, but may not have become stuck to that band. When counting spotted lanternfly on the sticky bands in the lab, we separated our tallies based on adults and instars. Unfortunately, we did not separate instars out into first through fourth, but looking back, we should have done that. We knew the percent of adults that escaped from those web coat sticky bands in that trial, and it was about 84%. And we took the average of the escape probabilities for instars 2 through 4, and that averaged out to about 31% from that trial. And so we adjusted the total count for each tree over the entire season. I might refer to this as number of spotted lanternfly for simplicity's sake, but it is that estimated value of the spotted lanternfly that encountered the bands based on the adjusted values. Since the number of days that each band was left out varied based on site, for each tree, we relativized the number of individuals encountering the band by dividing that value by the number of days the tree was actually banded. Last little bit of methods. When we were banding trees, we took care to include tree species other than just tree of heaven, though we did target tree of heaven. We selected trees by laying out 2 transects and using a random number generator to select the coordinate and then banding the tree closest to that coordinate. So we had species other than just tree of heaven. And so the first question we wanted to ask using our new estimate of daily SLF counts is, does tree species affect the daily number of SLF encountering a tree's band? Our dependent variable in this case was the total daily estimated number of SLF. And these data were pulled across 19 sites, and a one-way ANOVA was performed on LN transformed data. We expected, just anecdotally, that our highest spotted lanternfly numbers would be on tree of heaven. And what we found was that the daily number of individuals encountering a tree band did vary as a function of species, but the differences we saw were between Ailanthus and red maple, with Ailanthus having higher daily encounter rates. Of course, by looking at these confidence intervals, you can tell there's a lot of variation in some of these groups. And this is due in large part to small sample sizes in some of our species. The hickories, for example, are a really good example of this. And so we actually have collected additional data that are being processed right now in hopes of elucidating some of this pattern and maybe getting some smaller confidence intervals to start picking apart true differences in SLF counts per species. I did break out the data for only red maple and tree of heaven from this data set, and I compared the mean number of individuals between those 2 species and did find that the inferred daily encounter rate for tree of heaven was just over double that found in red maple. The next question we asked, is there a correlation between a tree's DBH, diameter at breast height, and the daily number of spotted lanternfly encountering that band? Because this answer could be very dependent on life stage of the spotted lanternfly, we split the analysis into 2. The first correlation dealt only with the number of instars encountering the band and DBH, and the second correlation dealt with the number of adults and DBH. And we used Spearman rank correlations for both cases. And what we found was for both instars and adults, there seems to be a positive yet weak correlation between DBH and the daily number of either instars or adults encountering that band. So we saw that DBH and number of spotted lanternfly encountering the bands were weakly correlated. We now wondered if the effect of DBH on the number of spotted lanternfly per day differed based on species. And so we ran an analysis of covariance on the pooled data to see if the effect of DBH on the number of spotted lanternfly per tree per day differs depending on species. And we found that the effect of DBH on number of spotted lanternfly per day does differ based on species. And so we had to break that further apart. And for each species that were included in this analysis of covariance, we ran separate regressions with the number of spotted lanternfly per day regressed over DBH. And I'm just showing a few of these examples because I'm running quickly out of time. That stuck out. There were similar tree species that exhibited similar patterns. Some of those included, I think, birch and box elder, if I'm not mistaken. So for each of these three species, we have Norway maple and tree of heaven and red maple. We see that the number of spotted lanternfly per day does increase as a function of DBH. But it should be said that this trend was not observed for all the tree species that we encountered. And so we're continuing work on this project throughout the spring and early summer. We recently incorporated newly collected forest inventory style data for 10 additional sites into our study. And we're now incorporating new spotted lanternfly counts from trees that were banded on these 10 sites into a larger data set. And we are continuing to focus on the tree layer, but also saplings, seedlings, and invasives across 29 whole sites using the full smattering of variables that we were able to collect. And we hope to use this FIA style data we collected along with true FIA data to draw some inferences about characteristics of infested forest sites. One of the big goals of our project that I didn't talk about today was to assess the damage that's being done by these spotted lanternfly. And so we have scheduled post-infestation revisits to each one of these 29 sites in order to assess current spotted lanternfly numbers using sticky bands, to assess damage and mortality in both the saplings and the trees that we previously measured in those sites, and to look at mortality and changes in composition in the seedling layer following infestation. So hopefully next year we'll be able to report on some of these measurable effects of spotted lanternfly in these forest strata. So I'd like to obviously thank APHIS and Westchester University, all the undergraduate students who have worked with, and the 29 landowners and land managers who've had the pleasure of working with. So thank you.
All right, thanks, Jen. That was great. I think there's a couple questions in the Q&A. So it was asked, was the tape reset after each individual SLF, or the tape trap, sorry, reset after each? And
yeah, that's a really great question. And I'm assuming that's talking about the experimental trial, and the answer was yes. So with every single trial, we actually had multiple wooden slabs so that we didn't use the same one every single time. We replaced the specific band of tape regardless of whether or not it was duct tape or sticky bands. We replaced it every single time, and we alternated whether we used sticky bands or duct tape for every single spotted lanternfly.
And I think you had this in your slides, but you can still answer this. Besides tree of heaven, were there any other species of trees used for this experiment?
Yeah, and just off the top of my head, so because we randomized the trees we were collecting information on, we had a lot of ailanthus, a lot of red maple, we had box elder, we had hickory species, black walnut, birch species, and I have them separated out by species, but I've compiled them just in Betula spa, for example. Gosh, what else? Sycamore, which Tracy Lesky mentioned something about sycamore. We do have some banding data for the sycamore, and I'd be happy to share that. There were several other species. I just don't remember off the top of my head what they were.
Yeah, I would just guess maybe there was cherry in there, but I don't know.
You know what? That's a very good guess. I think we did have very few cherry. We had some, very few, and we actually had some oaks. So.
And this is a question. Do you have any data on double sticky banding?
I don't.
Okay.
Sorry.
Oh, no, it's okay. I was yeah, I think I understand what the question was about.
I took it as double sticky banding, like double in width. Is that what the?
Yeah, well, I've seen it too, where it's been put on trees where it was like there was one band and then a second band, like there was
Right.
yeah, or larger, or like you said, they were stacked and maybe they had a larger distance across or something.
Yeah, I don't. And the reason for that is we always chicken wired our sticky bands.
Yeah.
And we pre-cut our chicken wire for sticky band width. So that's why we didn't do any of that. And because sticky bands are expensive.
Yeah. And this is another question. They were thanking you for your great presentation, but just wondering, how do you decide the length of escape window? And will you consider adjusting the time period depending on day-night captures?
So we think about the escape window for the experimental trial, I'm assuming. The way we determined that, I mean, we really had to sacrifice some spotted lanternfly, which I've killed enough in my lifetime now. We literally let them crawl onto each one of the types of bands. And for the first, 25, 30 individuals, we would just sit there until the deed was done, and time how long it took. And we basically figured out, I think we did a regression and figured out that if they have not escaped in 2 minutes, they never escape again.
Okay. And would you consider adjusting the time period depending on day-night captures?
I honestly hadn't thought about that.
Okay, I know, that's okay. And then this was, a question which you probably did think about this, because I know other people have tried this too, but did you consider tanglefoot applied to tree wrap paper?
Yeah, and I think the reason we kind of did it the way we did is because it was incredibly opportunistic. Like, we just happened to be out in the woods with our sticky bands, and we were like, okay, if we collect insects right now, how is this going to help in the numbers that we are collecting? So that's the opportunistic nature of this really precluded a lot of other methods, unfortunately.
Yeah, I know from other people that looked at different ways to capture them, that the tanglefoot was pretty messy, but also like and most of the time the bands are being brought back to do counts, and that was really difficult with something treated with tangles, because everything would all stick together.
Yeah.
So Greg, this is Kelly. I can't type into the Q&A for some reason because I was
You're a panelist. You're right.
So my question for Jennifer, hey Jennifer, is that did you ever try bug barrier? We've had much better success with that than with web coat. It does decline a little in force in adults and efficacy, but it was a lot better than web, and you don't have to worry about the non-targets. Yeah,
so we have used that. I think more in demonstrations. The draw for the web coat, just basic sticky bands for us, was really the cost and ease, because we were working across 19 sites, average of probably 15 trees per site, and changing bands and counting every 2 weeks, and it was just a lot of work. And so anything that made it a little quicker and cheaper was what we were going for. But yeah, since this, I have been looking into other trap types, and just the bycatch difference alone draws me toward those for not a huge quantity of trees.
Have you tried to talk to the owner of the company that sells bugberry? Because you can get it at a discount as a researcher. Send me an email and I'll send you its email address and phone number.
Okay, thank you so much, Kelly. I appreciate it.
You bet.
And there was one other question that came up, Jen. Did you observe any avoidance behaviors? And their comment was, in the field, they've noticed adults using their front legs to test the surface, and then not advancing onto the sticky surface.
Yes, those are the vein of my existence. And yes, we noticed avoidance behaviors both in the field and in the lab. And so I didn't mention it in this presentation, but something else we experimented on during the trials was not only the life stage and not only the band type, but we also looked at the number of legs that became initially stuck. Like as they walked onto it, the number of legs that became stuck before they attempted their escape, to see if that made a difference in the proportion of individuals that were able to escape. And we did see that as the number of legs initially encountering the sticky band increased, the escape probability decreased.
Okay. Yeah, and I think it was Tracy Lesky in her lab did some work also where they found it didn't take very much for them to get what they're describing as occlusion, where you only have like 25 to 30% of the band with SLF on it, and they could just bypass the band basically because of the numbers of SLF on the band. So that was another issue.
And we found something similar. I had a GIF of it and I was going to put it in the presentation, but I didn't really know where it would go. One of the other issues we had with using sticky bands, because we refused to use them without chicken wire, is that the adults especially, and also for thin stars, they crawl right over the chicken wire. It's like this, either they use a carcass bridge or they use the chicken wire to often get over the sticky band. So that's something that's not quantified.
Okay, all right. And I don't see anything else in the questions and answers, but there might be something in the chat if you want to take a look at that too.
Okay.
So thanks again for your presentation, and then also to Melody and Houping.
Thank you.
So